Let's start with dendritic cells. Among all antigen-presenting cells, the most potent ones are the dendritic cells. And they are widely recognized as the most potent APCs, due to their unique ability to efficiently capture, process, and present antigens to T cells, thus bridging the innate and adaptive arms of the immune system. As these dendritic cells are the part of first line of defense in the immune system, which is body's immediate and nonspecific response to invaders. And the dendritic cells are strategically located in the tissues and organs throughout the body, including the skin, mucous membranes, and lymphoid tissues. And thus, they are among the first immune cells to encounter invading pathogens, toxins, or any danger signals. When dendritic cells encounter these foreign substances, they capture them, then process these antigens, and then migrate to the draining lymph nodes, where they present the antigens to T cells and B cells, which are the important components of adaptive immune system. So, this interaction initiates specific adaptive immune responses, which are the body's second line of defense. So, dendritic cells are part of innate immunity, but they capture and present antigen to the components of adaptive immunity, and in this way activate them to initiate adaptive immune response. That's why we say they act as a bridge between innate and adaptive immune responses. Morphologically, dendritic cells exhibit a distinct appearance, characterized by their long, branching surface projections called dendrites. And that is the reason why they are named dendritic cells. These dendrites significantly increase the surface area of the cell, facilitating antigen capture and interactions with other immune cells. Also, these dendritic cells typically have a larger cytoplasmic volume compared to other immune cells like lymphocytes. And this increased cytoplasmic area allows dendritic cells to accommodate the large number of organelles involved in antigen processing and presentation. And we know these dendritic cells are strategically located in various tissues and organs, where they constantly survey the environment for potential threats. They are particularly abundant in the skin, mucosal linings of the respiratory and gastrointestinal tracts, and lymphoid tissues, such as lymph nodes and spleen. One of the key features of dendritic cells is their exceptional antigen capturing capacity. And this capacity is due to a specialized feature. Dendritic cells possess an array of specialized receptors, including pattern recognition receptors or PRRs, such as toll-like receptors or TLRs, C-type lectin receptors or CLRs, and scavenger receptors. These receptors enable DCs to recognize and bind to a wide range of pathogen-associated molecular patterns, PAMPs, and damage-associated molecular patterns, DAMPs, present on pathogens or dying cells. Well, what are these pathogen-associated and damage-associated molecular patterns? That are PAMPs and DAMPs. These are actually two important classes of molecules that play a role in initiation of immune response, as they are recognized by the immune system as danger signals, alerting it to the presence of potential threats and tissue damage. Pathogen-associated molecular patterns, or PAMPs, are the molecular structures that are conserved among groups of pathogens, such as bacteria, viruses, fungi, and parasites. And these structures are essential for the pathogen's survival and are not typically found in the host organism. So, when the immune system detects PAMPs, it recognizes them as indicators of infection and triggers an immune response to eliminate the invading pathogens. For example, the bacterial lipopolysaccharides are found in the outer membrane of gram-negative bacteria only, and not in any other form or in host cells. So, they then act as PAMPs for immune cells recognition. Then, DAMPs, on the other hand, are endogenous molecules that are released or exposed by damaged or stressed cells. They then serve as alarms and indicate tissue injury or cell death. So, as PAMPs alert immune system about infectious dangers. Similarly, DAMPs play a vital role in alerting the immune system to non-infectious dangers, such as tissue damage, trauma, and cellular stress. So, the PRRs that are found over the surface of immune cells, mainly dendritic cells, recognize these PAMPs and DAMPs as alarms and initiate a signaling cascade that triggers immune response against the exogenous and endogenous harms to the body. Now, let's look at this mechanism of antigen recognition and capturing by dendritic cells. So, what happens is that upon encountering pathogens or foreign substances, dendritic cells initiate several processes to capture antigens. Such as, they can utilize phagocytosis, receptor-mediated endocytosis, or micropinocytosis, to internalize and engulf pathogens or antigenic material. Where we know the ultimate outcome of all these three processes is same. That is the engulfment of antigen inside the dendritic cell. But their mechanism may vary a bit. Such as, we know that phagocytosis is a cellular process adopted by various professional phagocytic cells, including dendritic cells. By this process, they can engulf and internalize large particles such as pathogens, cellular debris, or foreign substances. Phagocytosis starts with the recognition of the target particle by receptors on the cell surface. These receptors can be specific for pathogen-associated molecular patterns, PAMPs. And we know PAMPs are the molecules that tag pathogens for destruction. So once recognized, the cell extends pseudopodia around the particle, enclosing it within a vesicle made of cell membrane. And this vesicle is called phagosome. The phagosome then fuses with lysosomes, that is an important organelle, containing plenty of degrading enzymes. This fusion then form another vesicle called phagolysosome. Then inside this phagolysosome, the ingested particle is degraded by lysosomal enzymes. And in this way, they get captured and processed inside the dendritic cell. Another mechanism adopted by dendritic cells to capture antigen is receptor-mediated endocytosis. Receptor-mediated endocytosis is a selective and highly specific process used by cells to internalize specific molecules or ligands from the extracellular environment. This process is essential for the uptake of various nutrients and regulatory molecules. Receptor-mediated endocytosis begins with the binding of specific ligands to their corresponding cell surface receptors, which are usually C-type lectin receptors or CLRs. These ligands can be various molecules, including hormones, growth factors, enzymes, and cholesterol. This ligand-receptor complex then gets enclosed in a clathrin-coated vesicle, pinching off from the cell membrane. This clathrin is a protein that helps invagination of the cell membrane. This endosome formed then gets uncoated and later fuses with lysosomes, where the ligand gets degraded by the lysosomal enzymes. In many cases, the internalized receptor can be recycled back to the cell surface for reuse, or it may also be degraded by lysosomal enzymes. Lastly comes the third process, for antigen capturing by dendritic cells, that is macropinocytosis. Macropinocytosis is a non-selective process by which cells internalize large volumes of fluid and small particles from the extracellular environment. So, we can say, macropinocytosis is a mechanism that enables dendritic cells to sample their environment, take up fluid, and capture antigens in a non-selective manner. So, macropinocytosis is particularly important for dendritic cells in their ability to capture soluble antigens and large volumes of extracellular fluid. So, we can say that dendritic cells have evolved to utilize all three mechanisms to capture and process antigens efficiently. By any of these mechanisms, the antigen gets captured by the dendritic cells and then processed inside the cell, just as we have seen in these mechanisms. And the purpose of this processing of antigen is to convert it into smaller peptide fragments. So, what happens is that within the endosomes or phagosomes, antigens undergo degradation through the action of proteases and other enzymes that enters the vesicle by the fusion of lysosome. This proteolytic degradation generates peptide fragments. These fragments can bind to specialized molecules called major histocompatibility complex or MHC molecules, which are synthesized within endoplasmic reticulum. And these MHC molecules are divided into two main classes, MHC class I and MHC class II. But dendritic cells predominantly present antigens to T cells through MHC class II molecules. The processed peptide fragments are loaded onto MHC class II molecules within specialized compartments called endolysosomes, formed by the fusion of endosome and lysosomes. This process is facilitated by various accessory proteins. We have explained this loading of peptides on MHC molecules in our other videos, under the title MHC and Antigen Presentation. You can watch it there for better understanding. Now, once the antigen-derived peptide fragments are loaded onto MHC class II molecules, they are transported to the cell surface, where they are presented to T cells. Dendritic cells express high levels of MHC class II molecules on their surface, which ensures efficient presentation of antigens to T cells. In addition to MHC class II molecules, dendritic cells also express co-stimulatory molecules, including CD80, also named B7-1, and CD86, also called B7-2. And these co-stimulatory molecules interact with their respective receptors on the surface of T cells, that are CD28 and CTLA4, respectively. These co-stimulatory signals are essential for the activation and differentiation of T cells. This interaction of dendritic cells with T cells usually occurs in lymph nodes that lead to T cell activation and differentiation, where the engagement of T cell receptors or TCRs on T cells with the antigen MHC class II complex on dendritic cells triggers intracellular signaling cascades, resulting in T cell activation. But this primary signal is not enough to produce an appropriate response. So, co-stimulatory molecules on dendritic cells, such as CD80 and CD86, engage with their receptors on T cells, providing necessary secondary signals for optimal T cell activation. Now, once the T cells are properly activated, then this activation of T cells by dendritic cells leads to a variety of outcomes, including T cell proliferation, differentiation into effector T cells, and the development of immune memory. In effector T-cell differentiation, dendritic cells are particularly effective at priming and polarizing naive T-cells toward specific subsets, such as T helper cells like Th1, Th2, Th17, or regulatory T-cells, depending on the cytokines and signals present during the interaction. So, in this way, dendritic cells present antigen to T-cells. And then T-cells process against it, leading to the fate of that antigen.
Introducing sqadia.com on your mobile. Making medical education more accessible than ever. Your one-stop shop for medical learning now fits in your pocket. Visualize, understand, and master as one seamless process. Access exclusive courses on anatomy, physiology, and more, right from your phone. With intuitive navigation and engaging video content, your learning becomes enjoyable and effective. Get started with our free trial. An abundance of educational resources is now just a tap away. Download now the sqadia.com app from Google Play or the Apple App Store. sqadia.com. Medical education at your fingertips.